All right, let's put on our detective hats today. We're going to follow a team of researchers as they use a pretty special kind of AI to crack one of the biggest, most frustrating mysteries in modern medicine. So, here's the mystery, the central question that keeps scientists up at night and costs billions of dollars. Why do so many drugs that look absolutely fantastic in the lab just fail when it really counts? And the scale of this problem? It's just staggering. Between the years 2000 and 2015, a whopping 86% of all drug candidates failed to get approved. Think about that. That's almost 9 out of every 10 drugs. And so often, the reason for that failure comes down to one critical unknown. And that unknown is the drug's true mode of action, or MOA for short. You can think of it like this. Scientists find a key that seems to help with a disease, but they have no idea which lock it's actually turning inside the body, or what other doors it might be accidentally unlocking along the way. This whole black box problem is just a massive roadblock, especially for these absolutely devastating illnesses like Huntington's disease. For years, scientists have found compounds that seem to help, but not one has ever succeeded in a clinical trial. To find a cure, the researchers knew they had to stop guessing. They had to figure out what these drugs were really doing inside our cells. So how do you even begin to crack a case like this? Well, to solve the mystery, the scientists basically became forensic investigators. They started gathering every possible clue from the cellular crime scene after a drug was introduced. They used this really powerful technique called multi-omics, which, yeah, sounds super fancy, but it's just their way of saying they collected 4 different kinds of evidence. They asked, what genes are getting switched on or off? What kind of fuel are the cells using? Which cellular machines are actually running? And what are the master switches controlling the whole operation? So here's a look at their game plan. They started with over 30 compounds that were known to have some kind of protective effect. From there, they narrowed it down to the 14 most promising suspects, and that's when they unleashed their multi-omics analysis on them, creating this absolute mountain of data, a pile of clues so huge it would be impossible for any human to sort through on their own. And this is where the hero of our story comes in. A special kind of artificial intelligence, a digital detective, specifically designed to find the hidden patterns in that mountain of data. Now, what makes this AI so special is that it wasn't your typical black box. This was an interpretable model. That means it doesn't just spit out an answer and say, trust me. No, it actually shows its work. It lays out the entire deductive process so the researchers can see exactly how it solved the case. And this is where the case took its first big twist. Because when the AI started analyzing all those clues, it found something completely unexpected. At first, things looked like a mess. When the AI looked at the first type of evidence, the gene expression data, basically the RNA fingerprints, it just saw a random jumble. The drugs didn't seem to have anything in common. It looked like a total dead end.
But then came the aha moment. When the AI analyzed a different set of clues, the metabolic data, the stuff showing what fuel the cells were using, a stunningly clear pattern emerged. Suddenly, the compounds just snapped into 2 distinct, unrelated groups, which the scientists called Group A and Group B. And this created a huge paradox. 2 of the drugs in the study, cyproheptadine and meclizine, are both common antihistamines. You'd expect them to be partners in crime, right? To work in similar ways. But nope. The AI placed them in completely different gangs on opposite sides of the map.
This became the new mystery. So how on earth could 2 drugs, both known for blocking histamine, be having such wildly different effects inside these brain cells? That was the big question the digital detective had to answer. So what did the AI find? It crunched all 4 types of data together, mapping everything onto this massive network of how molecules interact. It was finally time to unmask these 2 mysterious groups. The AI's network analysis revealed Group A's secret mission. You can see it in the highlighted parts here. These compounds were all involved in a process called autophagy. And that's essentially the cell's internal cleanup and recycling crew, the team responsible for getting rid of damaged parts. And what about Group B? Well, the AI found they had a totally different mode of action. They were messing with the cell's power grid, its bioenergetics. Specifically, they were going after the electron transport chain, which is a key part of how our cells make energy. Okay, a deduction from an AI is one thing, but you need proof, right? So the researchers went back to the lab, and the results were incredible. Just look at this. On the left, you see cells treated with Group A drugs. They're lit up with all those green dots. That's a clear sign the cleanup crew is hard at work. Now look to the right, at the cells treated with Group B drugs. Nothing. The AI was dead on. And they did the same for the bioenergetics claim. This chart shows the rate of energy production from the cell's little power plants. And would you look at this? The two groups have the exact opposite effect. Group A compounds actually increased energy production, while Group B compounds decreased it. Once again, the AI had cracked the case. The lead researchers really sums it up perfectly in their paper with this quote. It means that just because you know a drug's official target, like the histamine receptor for our two antihistamines, that doesn't mean you have any idea what the whole story is. So what's the big takeaway here? This investigation proves that a drug's known target is often just the tip of the iceberg. It shows that by combining different types of biological data, that multi-omics approach, and using an AI that can actually explain its reasoning, we can finally open that black box and see what's really going on. This could accelerate the hunt for cures for countless other diseases. And that leaves us with one final, really tantalizing thought. This was just for Huntington's. What other medical mysteries could we solve? What other cures could we finally discover if we could see what all of our medicines are really doing inside us?